Okay, well, welcome back. We have been this month talking about the series we've been discussing for our professional development Thursdays for
Yes.
inspiring classroom, inspiring ideas for the classroom. We've been doing, as hopefully you've been following along. If not, go listen to them and come back. And last time was questioning, right?
It was.
All right, okay. And
Yeah.
all right. So for this time, we've expanded the crew. Bill's joining us, Bill Bailey. And so we are going into what's next after we have that question. What's next? And so we get the kids to come up with questions. We figure out which ones are searchable, which ones aren't open ended, closed ended, and talk about which ones we can research. And so then, we narrow it down, find out what's feasible, and which question are we going to research. So now we have that question. So what are we going to do? And so obviously we make a hypothesis, right?
We
Makes
do.
sense. Makes sense.
Yes.
Make a hypothesis, right? And so we're going to, here in a second, we'll go through it at the different levels, the middle school or the elementary, middle, and high. The three of us, each of us is going to take a level and go over how we would explain this to students and how we would kind of walk through examples. We have examples for those levels too. And so hopefully this is helpful when getting the kids to go from having that question to go from there to go to actually starting planning their test because we want to do some sort of test to collect data. And so this hopefully is that gets you from the question to actually starting the test. And so, of course, if we pull up, if we stick with the theme, and say, Sarah, I think you said we need to stick with that theme with the
Stick
globe
with our theme.
cluster.
Yeah. We like that globe scientific process in lieu of the scientific method. And we've kind of been going each week on the I think we started just to have a starting point with the observing nature. And then last week we moved to the questioning. And now this week we're going to take a look at that. Planning and conducting an investigation. So looking at the different variables that we're going to use. And then once we've collected that data, look at analyzing and interpreting the data. So creating tables, graphs, and charts to both illustrate and summarize what the data that we are collecting.
And so what we want to do is we're going to go through, and I'm going to start with elementary and go through with the elementary one. Let me do a share screen real fast. And did that pop up?
It did.
Woohoo! All right. And so with the share screen, here's the infographic for the elementary side that we would use when we're going from our question. And we said we start there at the top, let's test our question. And so if we had that list of questions and we think we wanted to use the three of us when we were talking the bottle biology, right? And so if we were to take in, if you haven't seen that, it's simply taking a bottle. Of course, I don't know if I can even see myself. Can you guys see my bottle?
Yeah. Okay. Well, I can see it, but yeah, you can because our tiles are at the top.
Okay. But so you just take a bottle, you cut the top third of it off, and some of the different ways of doing it. Some of them you just put slots in it and slide it down. Other ways you drill a hole in the cap and you turn it over. And so there's different ways of doing those, and we will have links for each of the levels, the bands, elementary, middle, high, each of the bands, the links that we had that we liked. That each of us picked a link that we liked for our level, and so the one we have for elementary, correct me if I'm wrong, is the globe one.
It is the globe.
Yeah. And so we'll have links to a way of doing those. There's not the way of doing any of those. There's hundreds of different ways, but it's a good way. So the question we're picking for mine, does the light make my plant grow? And so, I mean, I've heard light makes plant grows, but does light make my plant grow is my question. And so then what I like to with our younger audience is we're going to take that question, we're going to guess what the answer would be. We're going to guess what that answer might be. And so what I put in my little think box there is I think, and I like using with our younger crowd, the I think for our hypothesis. We're going to put it in as an I think. And so I think the more light my plant gets, the faster it'll grow. And so they might start off with, does the light make my plant grow? Of course, that's a yes or no, isn't it? And so we want to change that into an open-ended, and so, okay, if we change it into an open-ended, that means something we can research, right? It's not just a yes or no. All right, and so will it grow fast? Okay, maybe. So I think light will make my plant grow faster, and so that's going to be what we call hypothesis. That yes is actually going to be a hypothesis, and we have in there a hypothesis is a testable, so we have to be able to test it. We have to test our idea. It has to be measurable. Oh, we have to be able to measure something. We can do that. And then it says it's intended to address the question. It means we're going to try to answer our question, right? And so we could see if it grows fast, we could maybe make we can test and we can measure to see how it's growing every day or something, and you see how fast it's growing. But all right, then we have next, let's test our guess. So we have to come up with one. But remember, it needs to be a fair test. And when we say fair test, means we can only test one thing, which is in science we're calling a variable, a time. And so when I say a variable, so we're just testing one type of thing. We can't see if we're saying light grows, then give one plant more water than another plant. It'd have to be the same. That's something else we could test. Maybe down the road we would want to test if the more water it gets, does it grow faster? And so we want to make sure we give them the same. It has to be fair. So we just test one thing at a time. And so with that, we have what we call dependent and independent variables. Now, what I like to put, of course, the independent variables, one we have in blue there, and it says this is the one that you change or you control in experiment. And so for ours, we're going to change, if we're testing the amount of light, we're going to change how much light plants get. So we'll have like 2 bottle biology things, and we'll put some seeds in one and put one in. We have to figure out and decide as a group how much light we would want it. Maybe we were going to put it in a window all the time. That would get more light, or maybe we have a plant light over it. And the other one, maybe we're putting a dark corner or something. And so we have to decide how much light it gets. And then we'll have to have a control one, right? So one that does it is just normal. And so then our dependent variable is the thing that we don't really have control over, but it's what we're seeing to see. We want to see what changes, right? So what's going to change? Well, we think how fast the plant grows is what's going to change. So that's our dependent variable. Now, these are really tricky sometimes, and sometimes it's hard to remember which one of those they are. And so remember, we're going to change the independent variable, but we're going to see what happens for the dependent. And let's see how we did on that. Let's test these.
And Steve, one thing that we said when we were planning this was even the 3 of us who have been doing this for a long time, we even go, okay, which is which? And you got to stop and think for a minute. And sometimes even the teacher's going to have to stop and think, okay, which one's which?
Definitely.
Like me, you're going to get it wrong a lot, and that's okay. It's a learning process, and it takes a lot of practice. So we just keep practicing, and we keep trying to learn from our mistakes.
And the nice part is, if we're doing this as a group, like a class, in my lower grades, I like doing things as a class. We get to help each other. Mm-hmm. And so we get to help each other. And so we have 3 different tests we could do here, right? I've already made my I think statement. And so these are my hypotheses, right? So the first one, I think the taller a student is, the further they can jump. And so what would be my dependent variable? Remember, the dependent variable, that's what we want to see how it changes. Remember that in the graphic? What are we going to see how it changes? Well, we're going to see what changes is how far they jump. And what's the independent, or the one that we're controlling? Well, we're having ones of different height, right? Mm-hmm. And so the height of the student would be our independent, because that's the one that we're controlling. And yet the one that we want to see what happens is how far they jump.
Let's just clarify that too, that we aren't controlling how tall the students are. But what we are controlling are the students of different heights that we're having them test.
Yeah, we're picking. If we have all shorter kids, that's not going to do it. So we're picking which ones. So we're controlling which ones do our test.
Yes.
Okay. Is that fair?
Yeah.
All right. So next one, I think the steeper a ramp is, the further my Matchbox car would roll. And so I just put a board there for a ramp, and I put my Matchbox top of the. I think the steeper it is, the faster or the farther it's going to roll.
Farther.
Okay. And so our dependent variable, the dependent is the I want to see. So what are we trying to see on that? Well, I want to see how far it rolls, right? Let's see if I'm right. I put those in backwards. All right. I put those in backwards. And so that happens. Oh, yep. My dependent, how far the car rolls. And the independent, well, we gave that away already, didn't I? It's this how steep it is. But I think you guys caught that one. All right. So last one for my elementary. All right. I think the darker a shirt, the warmer it will be in the sun.
Mm-hmm.
So I'm thinking when we all go outside, the days I wear a dark shirt, it just seems like I feel warmer. And so I'm like, oh, maybe these shirts. So we could put shirts like a white shirt, so a really light colored one, and then like a middle colored, then a really dark one. We could take their temperatures and see. So when we say our dependent, that's the thing that we want to see how it changes. What do we want to see? Well, we want to see if it's warmer, right? And so the temperature of the shirt in the sun is what we want to see, right?
Mm-hmm.
We want to see the temperature because we have to have data to measure. So we'll measure it with the temperature. And so what's our independent variable, or the thing that we're going to control what changes, right? Is the darkness, how dark or light it is, I bet.
Yeah.
Yes, how dark the
Control
shirt.
what shirt you wear.
Yeah, we can control that. And so those are 3 examples of hypothesis and which one's dependent and independent. And so remember, always ask, for hypothesis, I ask, I think it'll do this. And if whatever you're trying to see what changes is our dependent. And then whatever we're controlling the change of is the independent. And all these little things, anything you could change is the variable. Anything we could change. All right, so we're going to jump up to we're going to jump up to middle school, where Bill's going to take over.
Perfect. So at middle school, and I love what Steve did there at elementary, at the end, depending on the level of the comfort level of the elementary teacher, they may only have one thing they're going to test for the entire class. So everybody's testing that same thing. That way, at the end, you can compare to other people, and the kids, they kind of know. And if the great thing in elementary, then if the kids mess up or something, they have other groups that have that same question and the same setup and everything, so they can look at their data at the end. Where at middle school, you might want to start branching out and letting the kids, you can still use the ecosystem in a bottle, the bottle biology, and you can have your bottle and you can have, your soil and your seeds or whatever you're going to have in there, but then let the kids come up with even more what they want to test. And at that point, what you want to also do with them is make sure that they're doing all the things that Steve was just talking about controlling the things you need to control. So making them justify why they're going to do the test a certain way and how it's going to be set up and what they're going to be looking at and how they're going to look at it. That to me is at the middle school level is great because it's their mind thinking through everything that they should be doing and how it should work. So, the whole idea of the independent variable affects the dependent variable. Middle school kids will want to test more than one thing at a time. They're going to try to do like 6, 7 things they want to be able to do. And you still got to keep them in control of going, no, you can only do the one thing, but I don't mind if you do something different, but then you got to tell me how you're going to make sure that nothing is going to affect your results because you have other variables that you've forgot about or didn't have as a part of it. So I just want to make sure that everybody gets that's what you're trying to do in the middle school level. Yes, I think we're going to probably do bigger level graphs, and we'll talk about that later, but and so collecting their data and what they do with the data is pretty important. So with that, let's go ahead and let's look at the 3 tests that they're going to do at the middle school level. So the first one, very simple. You want to compare brands of paper towel to see which holds the most liquid. But before we even talk about that, so that's one test that they came up with. Someone may say, well, I want to see how much weight it's going to hold, when it's wet. I mean, so these are all these things you could do different with this middle school level kids. But this is the one you want to compare the brands of paper towel to see which holds the most liquid. So going back to the dependent and independent variable. So if it's a dependent, it's how much liquid the paper towel absorbs. And then the independent is the brand, because that's really what we're going to try different brands. We have no idea how much liquid's it's going to hold. So that is dependent on the independent variable. So each one is going to be different. So it's going to depend on how they made the paper towel and how good of an absorbent it is, right? So, which I think Bounty's the quicker picker upper, right? Doesn't say it
It's
absorbs
going to be
more.
way more than the school issued brown paper towel.
The brown non-absorbent school paper
Right.
towel. Yes.
Yeah.
That stuff is,
Right.
that should be in trouble.
Yeah.
Okay. So test 2, you want to determine how far people can see into the infrared part of the spectrum. And what's a great thing is, using truly things in your classroom that you want kids to know about, then they can start to test and figure out as well. And this is definitely something in the middle school level you'd want to do. So when you're looking at that, the dependent variable is going to be whether the light is absorbed in the infrared part of the spectrum. Whether the light's absorbed. Observed. Oh, click again. Oh, okay. Whether the light is observed. Sorry. And then the independent is the wavelength of the light. So you're going to look at different wavelengths, and can you see it?
Mm-hmm.
And the other one is it observed. So, yep. So that's test 2. And then test 3, determine whether how long a student sleeps affects score.
Yeah, that's worded very awkwardly.
Yeah. Huh. Okay. Well, so you're looking at how long a student sleeps. If I sleep 8 hours, do I do better on a test? If I sleep 3 hours, if I don't have any sleep, do I do better on a test? So the dependent one is the test score. So you're looking at truly what's the test score going to be? Go ahead and click.
Oh, I was trying to rewrite that question in my head.
That's all right.
I'm like, that's worded weird.
And then the independent is the length of time you're sleeping. So that's something that we are going to control, how long people sleep, and then we'll see what happens with their test scores. And I got to, as we were talking about this earlier, going back to the bottle and looking at all that, even when we do this, you hear us talking through this in our head. This is what we want kids to do. That's the same way that we as teachers do. You start thinking about which one is the one that we're trying to look at, and then the effect of that is the dependent variable. The dependent variable is basically your result, like what happens after you do the test. What are you seeing? And so it's how much test score, how much liquid, whether the light's observed, all those things.
There we go. All right.
Okay.
Jumping over.
Yeah, we've taken this now from elementary, where it's possible you'll be looking at one investigation for the whole class, to middle level, where now we're going to open it up to student groups and have them take more ownership in the tests that they'll be doing and selecting their independent and dependent variables. And so when we get to high school, we expect them then to not only think of their question and their own hypothesis and identify the variable that they'll be using, but also that we expect it to be more technical, and that we expect quantitative data to back that up. So some way of collecting that data and then interpreting that data as well when we have that collected. And part of that then is going to be graphing the data that we get. So we will still identify the dependent and the independent variables, but then we've got to think about how are we going to be displaying this data once we have it. And we found this graphic that I think we all really liked this. We found it, it's at the bottom, I think it was ThoughtCo is where we found this, but this dry mix. And I'd never seen this, but I love this. This is always, I would have trouble remembering, okay, where do I graph this? Where does the dependent variable get graphed and the independent variable get graphed? And so you can see the acronym there that the dependent or responding variable is graphed on the y-axis, and that the independent variable, or the variable that you will manipulate, that you will control, is graphed on the x-axis. And so I like that just to help us remember the dry mix. And then also we came up with this graphic as well when you are graphing for the x and y axis, which is which. And I know we had a discussion yesterday, and Stephen came up with came up with this great X to the left, Y to the sky. And I like that he used that. And I think that would have been useful to my students too, had I known that, yeah, the Y, especially the Y to the sky part, right? That's the part that you're going to draw up and down, clear up to the sky, that's your Y axis. And then from dry mix, I know that DRY is going with my dependent variable. And then X to the left, if you start at the right and go to the left, and then there's your X axis. And with the mix part of dry mix, that would be your independent or manipulated variable graphed along the X axis. So if we advance the slide and we'll look at our potential hypotheses for high school level, and looking at these that would be appropriate for, like Bill was saying, something that you might be teaching as part of your class. I know, for example, colligative properties is something we would cover in high school chemistry. And so adding table salt to a beaker of distilled water will increase the boiling point of that water. That's something that we could test. And if we look at that, our dependent variable would be the temperature it takes to reach that boiling point. Our independent variable that we have is going to be the amount of salt that we're adding to the water that we're testing. But then beyond that, we're going to also start identifying what we're controlling. And the controlled variable in this case could be the amount of water that we're using. If we use more water in one beaker and less water in the other, then even if we're varying the different amounts of salt, there's other variables, working on that as well. Whereas if we're sure that the amount of water used across the board is consistent and that all we're changing is the amount of salt added, then that's truly what we're testing in that. If we look at test 2, I would always start the year with density in chemistry. That's something we would look at. And a penny minted in 1982 or earlier will consist of a higher percentage of copper than pennies minted after 1982. There's a lot of different ways that you could go about testing that. And a lot of students are able to find a couple pennies, that you can test. I was always collecting pennies for this. So if I think about my dependent variable, I'm looking at the percent of copper within those pennies. That is going to depend on the year that they were minted. So my independent variable for this is going to be the year that the penny was minted. And those are what I can't control when that penny was minted, but I can control the sample that I'm using. So I can choose pennies minted, just looking at the date. And then the control, again, there's going to be several controls in place, but one is that I'm using that same method to test the percent copper. And then if I look at my third example, water sampled from a shaded area will have a higher level of dissolved oxygen compared with water sampled from an area that has no shade. So for this one, my dependent variable is probably going to be my dissolved oxygen reading. My independent variable is going to be the place I'm choosing where I'm going for my water sample. But in that, this is, again, it's getting a little more complicated. When we're doing our sampling, something that we would want to control would be then the depth of water, and also that we're sampling water from the same source of water as well. And also that we're collecting water at the same time of day. So it does start to get a little more complicated as those tests that you're wanting to do also get more complicated, I guess, or more intense.
Yeah.
I don't know if we want to bring into it at the high school level then for those, the bottle biology or the terrariums that we're doing. Now we're just adding in another place where students can think about data that would be collected. What data could I collect from this? What is my question? How am I going to be able to research that question? What data am I going to be able to use to try to answer the question that I have?
Yeah, they're going more than just everybody planting radish seeds, or grass seed, and see which grows faster.
Mm-hmm.
And one thing that is so awesome is, and kids always ask, okay, so why do we got to control all this stuff? Why do we got to do all the, and eventually they go, well, okay, so if people are doing research and they really want to know, did this chemical or did this drug actually cut back cancer cells? Well, they better do it and have good steps on what they did to get to that point, so that it can be replicated by other people who can test it to see if it's actually replicatable other places. So can someone in Australia do this same test and everything works and it comes out the same way? And so we talk about that a lot with kids, just make sure that you understand that we're trying to eventually get you to the point where if you write procedures or you write whatever you're going to do, that it then can be tested by other people.
Absolutely.
All right, so we went through all three levels here on helping kids come up with the actual testable thing from their question. And as Bill mentioned early on, one of the important things after they do the test and they analyze their data is going to be communication. And so next Thursday's professional development will be on communication, where we're going to discuss different ways and techniques of allowing students to be able to communicate their results and findings and be able to share. And we'll discuss assessments and stuff like that for it too. So join us next week.
That's right.
Yeah, thank you. Say bye, Bill.
Bye, Bill.